Следующий доклад нашего коллеги из Германии, Фändrich. Превенция органной недостаточности с помощью торможения апоптозы. Возможности, перспективы использования в терапии. Ну, интригующий, конечно. Доклад должен быть. Пожалуйста.
Thank you. Okay. Well, ladies and gentlemen, thank you very much. First of all, I want to express my thanks to the Russian Academy of Science, which invited me to give this lecture. Maybe there's a psychological comment at the beginning of my talk. You all are aware that the first 20 seconds are the most important for a lecturer, because they decide whether people listen or don't listen. So, in order to get your full concentration and attention, I want to start my lecture with an example which illustrates why I think that the apoptotic mechanism and how we treat it may be of great importance in the field of medicine. Imagine you're sitting at home and you're suffering from acute chest pain. So these are symptoms of a heart infarction. And now, if this is the case, time is really running for you. Each minute you lose to get to the cardiologist and to get re-access in your vessels, re-dilated and reperfused, the risk that you will die from a myocardial infarction is increasing tremendously. So this is why I'm doing this work and why I try to show to you how important this can be. Apoptotic events play quite a role in a variety of different diseases. As I mentioned, it is important to block apoptotic cell death in the beginning of a myocardial infarction. This is also the case if you suffer from a stroke. It is very important to block apoptotic cell death in the first hours and to get re-access to reperfuse the tissue. And there are other examples, for instance, in infectious diseases. We have fulminant apoptotic death during hepatitis B virus infections, but also in trauma. I will give you an example at the end of my talk how apoptosis is affecting the injury of the spinal cord. And of course, we know a couple of different fulminant immune responses in autoimmune diseases where apoptotic events are important. So, what did we do? And what I'm presenting to you is an anti-apoptotic fusion protein. And this fusion protein consists basically of two parts. One part of the protein is derived from the HIV virus, from the AIDS virus. And this part describes a protein transduction domain. And this part of the protein will allow the whole protein to be inserted in an affected cell. The second part of the protein consists of a gene segment of the cowpox virus, the cytokine response modifier A part of the gene. And this part will block apoptotic events. So, just a few comments on the physiology of cell death in men. We know that there are basically three different pathways which can trigger and initiate apoptotic events. For instance, there's the extrinsic pathway. You know that there is a molecule called FAS, and this is a death ligand. a protein which can bind to the cell membrane, and then there is a death receptor and an adapter protein which oligomerizes and then gives rise to cleavage of caspase 8, and the cleavage of caspase 8 then activates caspase 3, and caspase 3 is an executor protein for cell death. So this is Fas ligand binding to Fas, initiating caspase 3 cell death. However, there's also an intrinsic pathway. Various stimuli like hypoxic stress, oxygen radicals, and other factors can initiate damage to mitochondrial homeostasis. And we know death signaling pathways which can impair the membrane potential of mitochondria, and then cytokine C is released from the mitochondria, and this also activates another caspase, caspase 9, which then also converges to activate caspase 3 and induces cell death. So this is the intrinsic pathway which activates mitochondria. The third one is mediated by the immune system. We know that, for instance, NK cells and cytotoxic T cells, they can release granules like perforin or granzyme B. And granzyme B by itself, when it's taken up by the cell, can directly activate caspase 9 and induce cell death. So these are the three different scenarios which we have to keep in mind if we want to block apoptosis. So, how does the fusion protein block these processes? The cytokine response modifier protein from this fusion protein blocks caspase 8, it blocks caspase 3, and it blocks caspase 9, as I will illustrate to you in a second. So if you are able to insert this protein into a cell, then this cell will, for a couple of hours, don't undergo apoptosis. And why did we choose the cowpox virus modifier protein? Because this protein is a highly evolutionary conserved protein. Over a million of years, it has been survived, and it has only one aim. This protein can only, or the virus can only survive in a healthy cell. And only in a living cell, the virus will propagate and replicate. So the virus, and especially this part of the virus, will do everything to keep a cell alive. And it has been published that this part of the cowpox virus blocks caspase 1, 3, 6, 7, 8, and 9. So this is not our work, this has been also illustrated by other people. So how do we get this part of the protein into the cell? As I told you, we have fused this viral part with the protein transduction domain and developed this fusion protein. And what happens is that this protein binds to the cell membrane, and then there is a pinocytose process which will pick up the protein, and the protein will be encapsulated in endosomes and then be released into the cell. And this shows you the experiment if we label either albumin as control or the fusion protein to a fluorescence dye and then give it to cells, you can see only the fusion protein was taken up by Jurkat cells in this experiment. And the uptake of the protein is a dose-dependent process. The higher the dose you give to the cells, the more protein goes into the cell. And it's a time-related thing. It's a very quick uptake within the first 30 hours after injection or giving into culture, you have the maximum uptake. So now I want to show you and demonstrate that actually in vitro first and then in vivo experiments, the fusion protein does inhibit apoptosis. Here we experimented in vitro. We did experiments with Urcut cells. And you see this is a normal Urcut cell population, and you don't find or only rare events of apoptosis in this cell line. However, if you treat these cells with an anti-Fas monoclonal antibody, then you see the apoptotic events which are tremendously increasing in this experiment. However, if you give at the same time the fusion protein plus the anti-Fas monoclonal antibody, you can significantly reduce the event of apoptosis. This actually shows a similar experiment where we used Urcut cells, and Urcut cells were treated with Fas antibody, and then the Fas mediated apoptosis of those cells. And you can see that after 5 and 7 hours of adding the Fas, you have the cleavage product of caspase 8, which we precipitated in Western blots. However, if we give the fusion protein, this cleavage, this caspase 8 activation is not visible within the first 5 hours. So the half-time of the protein is about 6 hours. This shows that caspase 3 is actively blocked by the protein. Normally caspase 3 metabolizes this substrate. So if you take Urcut cells and activate the Fas pathway, you can find this substrate after Fas activation in an increasing amount over 5 hours. However, this caspase 3 product is significantly reduced if you treat with the protein. So this gives you evidence that caspase 3 is actively suppressed by the protein. Now I'm going to show you that the mitochondrial apoptosis is also blocked, as shown here. Caspase A will activate a BID, which will then destabilize the mitochondria and give rise to cytochrome C. And there's a substance called staurosporine. Staurosporine will destabilize mitochondria directly, and we can show here that if you give staurosporine, then the membrane potential which we measured, the electronic potential of the membrane, was significantly reduced from normal. If we add the protein, we can restore this membrane function in the mitochondria as well. This is an illustration that caspase 9 is also saved from apoptotic activation. Here you see that if you give etoposide, which is another mitochondrial damaging component, this substance will damage the apoptotic membrane, and it will activate caspase 9 here. This is caspase 9 cleavage product after 3, 5, and 7 hours, and the protein can protect caspase 9 activation for the first 5 hours. So I have shown you that we can block caspase 8, caspase 9, caspase 3. So how does this translate into in vivo experiments? We injected a Fas antibody to animals and induced a multi-organ failure. You can see that if you inject 5 microgram of the Fas antibody, then all animals die within 7 hours after injection. However, if you pre-treat the animals 10 minutes before you give the Fas antibody, almost 90% of the animals survive. This is an illustration of the multi-organ failure, which we could demonstrate histologically in liver, in kidney, in small bowel. I want to make it more clear to you, picking the liver. You see that this is after 5 hours after giving the Fas antibody to the animal, and you see 8 hours later you have an absolute necrotic liver tissue. The liver cells, the hepatocytes are dead. However, treatment with the protein can prevent this acute apoptotic failure in the liver. And also the histology demonstrates to you that liver necrosis is evident after 8 hours, and you have more normal architectural portal fields and hepatocytes in comparison to control if you prevent this with the fusion protein. But this was given at the moment where you have the problem. Now I want to give you an example that you can also treat after you have the problem. So we injected or we ligated the coronary artery in mouse hearts and induced the infarction. And you see, this is the scarring in the heart after 1 hour ligation of the coronary artery. And you see a significantly better perfusion of the heart muscle if you prevent this by injecting this protein. And if you give the protein at the time of ligation, then you have a tremendously 40% less infarction size than in untreated animals, and the ejection fraction is significantly better. However, this was given here, the fusion protein was given before ischemia. If you do it 1 hour after ischemia, this would be the clinical relevant experiment. Then you see even better prevention of the heart muscle in comparison to untreated animals, and a significantly better ejection fraction in comparison to untreated animals. And I have a lot of experience in stem cell therapy, treating myocardial infarction and so on. And you can look up the literature, you will never find a stem cell experiment where the infarction size was significantly less than in untreated animals or patients. You can improve the ejection fraction with stem cells, but you can never improve the size of the infarction. So, last but not least, my last 2 minutes, it has been shown by this group from Anna Martin Villalba that neutralization of Fas ligand promotes regeneration and functional recovery after spinal cord injury. This has been published in Nature Medicine and is important to all acute trauma where people suffer from acute spinal cord injury. And here's the experiment and the data, very briefly. What they did in mice was they transected 80% of the spinal cord in these animals. And after transection, if they let the mice swim in water, you see the mice cannot move the tail, and they did not move the hind limbs, the hind legs. However, if you treat the mice within 1 week after the trauma, you have 1 week, then the paralysis is restored and they can move the tail and they swim again with the hind legs. They have the mobility. And why is this? This is a very important piece of information. I didn't know it either, but I looked it up. Once you have a spinal cord injury, within 6 to 8 hours after the injury, immune effector cells from the gut are activated and migrate to the spinal cord and do the injury and the damage to the tissue by a chronic inflammatory process, killing the microglia in the spinal cord. And this is a Fas ligand mediated process of apoptosis to the microglia in the spinal cord. And if you block this Fas ligand activation, which we can, then you can reinstall the mobility to the animals. So I think, as I showed to you, in all these indications of acute events, apoptosis is important and can be treated. What we have to do now, and this is what I'm trying to do now, is to produce the protein under GMP conditions on large scale and to perform a first clinical trial in cardiac disease in order to prevent myocardial infarction. And these are the people I have to acknowledge who have contributed to this work, especially my colleagues from the nephrology department and immunology department who have given me the support to present this data. Thank you very much.
Thank you very much. Ну вот времени нет, но невозможно. 2 вопроса, пожалуйста. Пожалуйста, да.
Thank you very much for your brilliant presentation. It is well known that DNA damage by itself could induce apoptotic cell death. Once you rescue such a cell, what would be the outcome of this cell? Because by preventing apoptosis, you could
Я прошу прощения, в микрофон говорить, пожалуйста, тогда и...
If you rescue such a cell, what would be the outcome? If you rescue such an apoptotic cell that potentially could bear a DNA damage, what would be the outcome for the whole organism?
Well, the point, this is a very important and critical question. The point is that the fusion protein, as I demonstrated in the kinetics, is biodegraded by the cell within 6 hours. So if you have a DNA damage and the protein is gone, the cell will die anyway because the damage will not be repaired. But if you have a hypoxic event, like in cardiac infarction or in stroke, there it is important to block this immediate acute damage, where the cell is still intact. In myocardial infarction, the cells are still intact, but apoptosis is induced via the intrinsic mitochondrial pathway. And this can be blocked for 5 hours about. And then the cell is rested. And in Germany, it takes about 2 hours from a patient, I checked that, when the patient has first symptoms, to go to the cardiologist and to be dilated. And still keep in mind, 50% of the damage to the heart takes place when the heart is reperfused. So after the occlusion is gone and the blood comes in again, then 50% are damaged. And this is an apoptotic event, and this can be blocked.
Пожалуйста, еще один вопрос. Ну, тогда короткий вопрос. Через какое время после начала болевого приступа? Через какое время после болевого приступа вы вводили препарат в больнице с инфарктом миокарда? What time was between the first cardiology and the infusion of your drug? Yeah, what time?
60 minutes. It was 60 minutes. Infusion
60 minutes? No, from cardiology from headache and
we
да.
ligated the artery, waited 60 minutes, and then we injected the protein.
Ну, то есть это как и сегодня принято в течение часа оказать помощь, да. Но результаты блестящие, конечно, да. В 2 раза уменьшение размера инфаркта миокарда, это, конечно, это здорово. Мы вас поздравляем. Профессор Левин просил один короткий ответ сделать.
К сожалению, вынужден уйти. Вопрос, где можно обучиться нашей технологии? В методическом нашем меморандуме есть адреса кафедры, где можно обучиться. И